Hi everyone. In this short video, I'll give you an overview of infections. We'll talk about the microorganisms that cause infections. We will look at how they do that, and we'll also try to understand some of the strategies that we can employ in order to prevent these infections or treat the infections once they are established in our bodies. As you all know, microbes or microorganisms are everywhere. They are all around us. The water we drink, the food we eat, the soil where we grow crops, they are all riddled with these tiny creatures. They are everywhere. And not all of them are harmful. Many of them are actually of good use to us. And one example is the normal flora, the organisms that live on the surface or inside our bodies. Many of them are bacteria, some viruses, some fungi as well. So they actually live in a harmonious relationship with us. They provide us with some benefits. So we provide them shelter and a good place to live, and they provide us in return. Some of them produce vitamins, some of them produce other nutrients, some of them help us break down certain molecules, and others, they actually prevent the growth of harmful organisms by creating a competition there. So they have a lot of benefits to us. At the same time, these actually can become problematic at times. In conditions where our immune system becomes weakened, they could cause opportunistic infections. So although they are friends, that they can become foe in cases where the immune system fails to keep them in check. How do microbes cause disease? We call them pathogens, the microbes which cause disease, and we will look at some of these features of a pathogen, like what is their mechanism of action, infectivity, pathogenicity, virulence, immunogenicity, and toxigenicity. And I will explain these individual terms in the coming slides. We can divide microorganisms into 5 broad categories: bacteria, viruses, fungi, protozoa, and parasitic worms or helminths. Towards the end of this lecture, we will look at strategies. How can we prevent these infections? We'll look at how vaccines work. We will look at what antimicrobial drugs or agents are available that we can use to fight these infections. And also we will talk a little bit about antimicrobial resistance. Some of these microorganisms have developed resistance to some of these drugs that we have. And towards the very end, we'll just mention a little bit about healthcare acquired infections, infections which are acquired in a healthcare setting, for example, in a hospital. The process of infection starts by colonization, which means these microorganisms somehow gain entry into our body and they colonize a part of the body. Yeah. From there, they establish themselves and they cause a local infection. Now, in order to prevent this, our immune system and inflammatory response will come into play and try to limit the spread of this infection. But if the immune system and inflammatory response fails, the infection can spread to other parts of the body. All right. Now moving on to the factors that I was mentioning to you earlier that cause these microorganisms to cause disease. So let me take you through each one of them. The mechanism of action is unique to each microorganism. Some microorganisms cause direct damage of cells. Others interfere with metabolic pathways in the cells. Still others produce toxins which are damaging to our cells. So different microorganisms will have different mechanisms of action. Infectivity is the ability of the pathogen to invade and multiply in our body. Okay, so for example, there are certain bacteria which produce certain enzymes which cause a protective layer to be formed around them, and they are able to cause infections. Pathogenicity is the ability of a microorganism to produce disease. A very simple definition. Virulence is how many pathogens are required to cause a disease. So if a microorganism is highly virulent, it means very small number of these microorganisms are able to cause disease. Okay, so they are more virulent. Immunogenicity is the ability of pathogens to induce an immune response. So certain pathogens are highly immunogenic, others are less immunogenic. And it's good to have pathogens which are highly immunogenic, which means your immune system can recognize them and mount a counterattack to get rid of them. Toxigenicity is a factor which is very important in determining a pathogen's virulence. And this actually talks about the ability of pathogen to produce soluble toxins, for example, endotoxin, yeah, mainly produced by gram-negative bacteria, as we will see in the coming few slides. Now, these pathogens are trying to invade our body structures, trying to colonize and cause infection. And what are we doing? Our body has its own defense mechanisms, right? So in order to prevent these organisms to invade our body, we have first line of defense, second line of defense, and third line of defense. The first line of defense is the physical barriers like skin, mucous membranes. There are enzymes present in the secretions which are produced by the mucous membranes. So all these are the first line of defense. Yeah. But if the organism is able to breach these, then we have our immune system to fight back. The first arm of the immune system is called innate immune system or innate immunity. This is the part of immune system which is always ready and it can mount an attack immediately. Then we have the third layer, which is acquired immunity. Acquired immunity is the part of immune system that has to first learn about the microorganism and then mount an attack. Now the good thing about acquired immunity is, or the response of acquired immunity is, that that response is very, very specific to the microorganism which is invading. It is slightly delayed, but once it's launched, it is very specific for the microorganism which is trying to invade. Let's move on to the classes of infectious microorganisms. So viruses, bacteria, fungi, protozoa, and helminths. Viruses are submicroscopic particles, and there was a time when they were even classified as dead objects, as dead particles, right? And actually they sit between dead and living. They don't have all the characteristics of a living organism. It's just a DNA or an RNA inside a shell. Bacteria, they don't have a nucleus, but they have other cellular organelles. They have all other features of a living cell. Then fungi and protozoa, all the way to helminths. Helminths are worms. They could be meters long and they could live in our body in, say, intestine, liver, etc., etc. All right, so these are the types of infectious microorganisms. So we'll start discussing bacteria. Now, bacteria, there are many different types, and most of them are harmless to us. But some are pathogenic and can cause serious diseases and even death. Classified based on the shape, so coccus or cocci are circular in shape, bacillus or bacilli are like a rod, and there are a few others. So coccobacilli, spirochetes, spirillum, vibrio, fusiform. So all these different types of bacteria are found in nature. One important thing about bacteria is the concept of gram staining. Now this stain is applied to bacteria and it causes them to pick up a dye. It will stain all bacteria purple, and then alcohol is applied. Some bacteria will lose the purple color, and when you apply the second dye, they will pick up red color. Okay, and the purple ones are called gram positive, and they have thick layer of peptidoglycan in their cell wall. That's why they keep that purple dye. The other type is gram negative. They have a thin peptidoglycan layer, but they have additional outer membrane which surrounds the cell wall. They are called gram negative. It is very important because the choice of antibiotic to treat these bacteria will depend on whether they are gram positive and gram negative. So this stain is a very basic diagnostic procedure when you're trying to identify the bacteria. All right, and this diagram actually shows you the difference between gram-positive and gram-negative bacteria. So as you can see that the gram-positive will pick this purple dye. It will stain the peptidoglycan layer. But in case of gram-negative, this layer is thin. They have this extra outer membrane. Yeah, the gram-negative bacteria. Now, bacteria produce toxins, and there are two types of toxins they produce: exotoxins and endotoxins. Now, exotoxins are usually produced by gram-positive and endotoxins usually by gram-negative. Now, the exotoxins are enzymes which are released during bacterial growth. So living bacteria can produce these and release outside. Examples are cytotoxins, neurotoxins, pneumotoxins, enterotoxins, and hemolysins. Actually, the name tells you what these toxins are doing. They can damage cell membrane, they can inhibit protein synthesis, and they are immunogenic, which means our immune system will produce an antitoxin against these exotoxins. The endotoxins, on the other hand, are basically released from dead bacteria. So when gram-negative bacteria die, their outer lipopolysaccharide layer actually releases these endotoxins. These endotoxins are pyrogenic, which means they will cause fever, and they lead to what is known as septic shock. Okay, so endotoxin, and a very small dose of endotoxin is capable of causing septic shock. And this is how endotoxins work. In this picture, it shows you that the endotoxin activates each and every aspect of our immune system and the inflammatory response. So it causes IgE antibody, which causes mast cell release. It activates platelets, activates clotting, it activates neutrophils, it activates monocytes, and you see a huge number of cytokines will be produced. So you have increased vascular permeability, leading to hypotension, causing shock. You have abnormal clotting. You have acute phase proteins being produced by the liver. You have TNF and IL-1 also acting on the liver, causing hypoglycemia and so on. So endotoxins have far-reaching effects, and they can cause septic shock, which, if not managed quickly, can lead to death. Bacteremia or septicemia are terms used to define presence of bacteria in the blood. Okay, and symptoms of gram-negative septic shock, as I was telling you earlier, is due to release of vasoactive peptides and cytokines that affect blood vessels, producing vasodilation. Blood pressure goes down. Oxygen delivery to tissues is not there. Okay, and gram-positive shock is also there, but the mechanisms are somewhat different. The symptoms could be very similar. All right, let's move on to viruses now. Now, viruses are, as I told you, submicroscopic organisms. So they are very simple microorganisms consisting of a nucleic acid, could be a DNA molecule or an RNA molecule, protected by a layer of protein around them, which is called a capsid. Now, these viruses or virions, the viral particles, can infect the cells, and they actually go inside and live in the cells. They cannot divide on their own. They need our cells to divide. So they use our cells as a factory. They go into the cell and they cause our cells to become their slaves. They use our cells' enzymatic machinery to replicate, and then millions of them will come out of the cell after replication. So either DNA or RNA, single or double-stranded, protein receptor binding site on the coat. Okay, when it goes inside the cell, it uncoats and it uses the machinery in our cells to replicate. The clinical symptoms will reflect the alteration of the function of the infected cell. For example, the influenza virus binds to a receptor on respiratory epithelial cells, causing symptoms of an upper respiratory infection. Yeah, very common. Now, this figure shows you what the virus does to our cells. The coat usually remains outside, and the viral genome will enter our cell. The viral protein synthesis will take place. These viral proteins can then assemble into a new virus, and millions of them, will leave the cell. They can cause, while they are replicating in the cell, cause metabolic derangements. They can cause cell lysis or cell fusion, and they can cause neoplastic transformation, which means this cell can become a cancer cell. So there are certain cancers, if you study their etiology, there is a viral infection. Okay, so a viral infection causing these cells to become cancerous down the road. Okay, so this is what I explained to you in the previous slide, but effects of viruses are listed here. They can inhibit host cell DNA, RNA, or protein synthesis, disruption of lysosomal membranes. They promote apoptosis, fusion of infected cells to adjacent host cells. So two cells can actually fuse together, become giant cells. Alteration of antigenic properties of host cells, and transformation of host cells into cancerous cells. They also promote, secondary bacterial infection. So if you have a viral infection, say sore throat, you are prone to develop a superadded bacterial infection. Fungi, the third type of microorganism. Now fungi are basically of two types, molds and yeasts. These are large eukaryotic microorganisms. They could exist as single cell, which is called yeast, or multi-celled, which are called molds. They adapt to host environment. They can survive in wide temperature variations. Some of them can digest keratin, which is the outermost layer of our skin, so they can invade. They can even live in low oxygen conditions. Okay, and they suppress the immune defenses in our body. Now, fungal infections are called mycoses. They can be superficial, like infection of the skin or nails. Okay, you must have heard of athlete's foot. They can cause deep infections, but deep infections are usually opportunistic, or they develop in people who are immunocompromised. Okay. Now, deep fungal infections are life-threatening and are commonly opportunistic. How do you diagnose these microorganisms in the lab? So there are many different ways to do that. Bacteria are detected from body fluids and specimens using cultures. Growth mediums such as agar promotes bacterial growth, and they can be incubated overnight in the incubator, and then you can view the colonies, and then you can take the colonies and spread them on a slide and stain the bacteria and observe them under the microscope. You can also do sensitivity analysis. For example, when you're growing bacteria in the agar gel, you put an antibiotic disc in there, and if the antibiotic causes the bacteria to die or cease growing, it means these bacteria are sensitive to this particular antibiotic, and that antibiotic will work in that patient. Okay. For viruses, mainly immunofluorescence. Serology can be done because the body will produce antibodies against the viruses, so we can detect those. Cell cultures is another way of detecting viruses. As a rule of thumb, viruses are harder to detect. The newer techniques which we employ these days to detect viruses include molecular techniques like PCR, where you amplify the DNA of the virus. If it is an RNA virus, you convert the RNA into DNA, and then you amplify the DNA, and you can detect this using PCR. For fungi, the mainstay of diagnosis is looking at the fungi under the microscope. They are very difficult to grow in a culture, so you look at them under the microscope. Infection control. Now, what do we mean by infection control? Infection control is both prevention and treatment. For prevention, the most important thing, and I think you all must be aware of this by now, the whole media across the globe, all healthcare advice is focused on these measures. Good hygiene, simple things like washing hands can make a huge difference. And if you have a patient who is infected with a contagious microorganism, you isolate them and you quarantine them. Yeah, so this is basically a mainstay of preventive measures. Vaccines are another tool to prevent infections. They induce long-lasting protective immune responses that will not result in disease in a healthy recipient. So basically, you use attenuated organisms or killed organisms or extracts of antigens or deactivated toxins, and you expose the person to these. The immune system will launch an attack against these. Now, these organisms that you're introducing to the person will not cause disease, but they will activate the immune system. So once the immune system is activated, the antibodies are produced. When the actual organisms attacks the person, these antibodies are there to mount an attack. But the challenge with vaccines is the development of a successful vaccine is costly, and it depends on several factors. You have to have, clinical trials, the safety of vaccines, efficacy of vaccines, and all that. We're living in this pandemic right now, and you must have heard a lot about these different terms and ideas. All right, treatment. How do we treat these infections? Now, infections can be treated by drugs. These are called antimicrobials. So you can have drugs against viruses, against bacteria, fungicidal drugs, antiparasitic drugs even. All right. Now this is just to give you an example of an antiviral drug, acyclovir, which is used for certain viral infections. Now, before I talk to you about this drug, there is no cure for viral diseases. There are some antiviral drugs. They do reduce the severity of disease, and sometimes they reduce the duration of the illness, but they are not curative. Yeah, we are still trying to find antiviral drugs which actually can cure the person. Anyway, what acyclovir does, it enters the host cell. It's converted to a form that can bind to the viral DNA. And once it's incorporated into the viral DNA, it prevents the replication of viral DNA. Okay. Antibacterial drugs are many, and they have different modes of action. Some of them will inhibit synthesis of bacterial cell wall. Others inhibit synthesis of nucleic acids. Some inhibit protein synthesis, and some modify metabolism of the bacteria. Now, the success of antibacterial drugs is because of the difference between bacterial cell and our cells. So our cells are eukaryotic, have nucleus. Bacterial cells are prokaryotic. So there are some fundamental differences between our cells and bacterial cells. So drugs that kill bacterial cells do not kill our body cells. Okay. Most of these antibacterial drugs are actually from other microorganisms. So the first antibiotic, penicillin, is actually, do you know where is it from? Which other microorganism produces penicillin? It comes from a fungus. So in natural environment, different microorganisms are competing with each other, and we can use some of those molecules to fight these microorganisms, right? Antibacterial drugs could be bacteriostatic or bactericidal, which means they can either stop the bacterial growth but not kill them, or it could be bactericidal. It will kill the bacteria. Antimicrobial resistance is becoming a big issue because of improper use of antibiotics and because of too much use of antibiotics. What happens, these bacteria which become resistant, they are able to inactivate the drug. They are able to reduce membrane permeability. They can alter the drug target on them. They can actively pump the drug out of them. They can transfer resistant genes to other bacteria. So they cross-communicate. And all these can be exacerbated by overuse of antimicrobials or failure to take full course. When you're overusing antimicrobials, right, you're killing your normal flora and you're providing an environment for bacteria to grow. When you fail to take full course, then you haven't killed the bacteria and you stop taking antibiotic. Those remaining bacteria have now seen the antibiotic and they will develop these features which will make them resistant to the antibiotic in future. The last topic I wanted to touch upon is healthcare acquired infections. So these infections are acquired by individuals during a stay in a healthcare setting, and in some cases may not become apparent until months after discharge from the hospital. Up to about 6% of hospitalized Australians are affected. Most common infections are urinary tract infections, surgical wound infections, and infections at the site of IV cannulas. MRSA is methicillin-resistant Staphylococcus aureus. It's a bacterium which is resistant to methicillin, which is an antibiotic, and MRSA is becoming a big problem. It is because of failure to follow infection control guidelines on hand hygiene, etc. So very simple procedures in the hospital setting, like good hand hygiene, can prevent emergence of strains like MRSA. Okay, and there are many other superbugs which are developing now because of some of our poor practices. So I think that's all for the topic, and I hope I was able to capture the infections and microorganisms in a nutshell.